Top 8 supplements that prevent and kill cancer. Cancer arises from the unfettered division of cells, a rebellion sparked by damaged DNA and exposure to carcinogens. However, research confirms that certain supplements can combat this growth by boosting protective compounds and signaling apoptosis, the death of malignant cells. This video will explore the top 8 supplements demonstrating efficacy against cancer development and progression through multiple mechanisms. We will overview key studies on each, their active compounds, and dosing considerations. Strengthening your defenses now is imperative, as cancer incidence continues to climb. Implementing these allies into a multifaceted lifestyle regimen can significantly deter onset and impede proliferation. While not panaceas alone, they are formidable co-therapies, suppressing factors that precipitate disease while optimizing bodily terrain resilience. Here are the top 8 supplements to help prevent and combat cancer. Vitamin D. This potent vitamin demonstrates anti-cancer properties through multiple mechanisms. First, it enhances apoptotic signals. Vitamin D communicates with receptors found in various tissues to regulate gene expression related to apoptosis, the programmed cell death that helps remove damaged or cancerous cells. Studies show vitamin D enhances signals urging these cells to self-destruct before proliferation worsens. Malignant cells often deactivate apoptosis, but vitamin D reactivates this protection. It restricts tumor blood vessels. Angiogenesis allows growing tumors to generate internal blood supplies, feeding their spread. Vitamin D significantly restricts the formation of new vascularity, according to recent McGill University findings, effectively helping starve malignancies by limiting nutrient availability. It regulates cell signaling. Uncontrolled signaling cascades enable cancer survival, division, and migration capacity. Vitamin D beneficially modulates signaling proteins like beta-catenin and hedgehog, which we've covered in past videos, restricting these pathways. It also upregulates tumor suppressor genes like P21 and P27 to hinder proliferation. It protects genomic integrity. The antioxidant activity of vitamin D defends DNA and cells from free radical damage that can spark mutation. Its anti-inflammatory functions also deter oxidative stress, enabling mutation. These preserve genomic integrity as a vital cancer prevention measure. It enhances immune surveillance. Vitamin D also activates key immune cells like macrophages and T cells that survey for threats like cancerous cells, priming attacks against early abnormal growths before malignancy develops. Deficiencies severely weaken this response. Vitamin D significantly reduces the incidence and mortality of numerous cancer varieties, especially breast, prostate, and colon. Findings unveiled at the 2018 Endocrine Society meeting that the top 25% of vitamin D levels correlated to a 31% lower risk of cancer-caused mortality over a 5-year period compared to the bottom 25%. Another study in the American Journal of Clinical Nutrition tracked postmenopausal women over 4 years. It revealed women deficient in vitamin D were 94% more likely to develop all-cause cancers. Yet another study showed that each 10 nanograms per milliliter rise in vitamin D levels equated to 15% less colorectal cancer risk. The collective benefits of fortifying levels are clear. While vitamin D is synthesized via sun exposure, oral supplementation up to 2000 IU daily is often necessary to reach blood levels between 60 to 80 nanograms per milliliter, where cancer protection and treatment benefits are maximized. This is especially true during the winter months or for indoor-oriented lifestyles in northern regions. Testing levels every 6 months monitors maintenance in this anti-cancer range. Vitamin C. Vitamin C demonstrates extensive anti-cancer impacts. It enhances immune function. Vitamin C concentrations tightly correlate with immune cell proliferation and efficiency at attacking threats like cancerous cells in the earliest abnormal outgrowth stages. White blood cells also require surging utilization of vitamin C when actively combating tumors and infected cells. Keeping cells saturated primes these defenses. It enhances antioxidant protection. As both water and fat soluble, vitamin C provides crucial antioxidant activity, defending cell membranes, proteins, and even DNA itself from free radical damage that can spark mutation. Its capacity for quenching inflammation and oxidative stress makes the terrain unfavorable for cancers getting initiated or advancing. It provides detoxification support. Vitamin C is needed for liver detoxification enzymes to function maximally, escorting carcinogens and toxins out of the body swiftly before they can damage DNA material. Chronic toxin exposures are huge co-conspirators with dietary and lifestyle factors, establishing the intracellular conditions enabling cancer development. Keeping cellular trash removal processes optimized is pivotal. It employs pro-oxidant selective effects. Intravenous vitamin C employs pro-oxidant mechanisms to generate hydrogen peroxide, which research confirms selectively destroys cancer cells. The reasons are multifold. Malignant cells tend to have higher iron levels that react with hydrogen peroxide and vitamin C to form cell-destroying free radicals. Also, tumors show less endogenous antioxidant capacity compared to healthy cells. Further, cancer cells harbor defective membranes, making them more vulnerable to destabilization from oxidative damage that vitamin C overwhelms their defenses with. It helps in restoring ECM integrity. Vitamin C is also essential for manufacturing collagen, the extracellular matrix essential for maintaining tissue integrity and structure. Tumors often have poorly structured, weakened extracellular environments and cell junctions that enable them to break through tissue borders during proliferation and metastasis. Fortifying ECM integrity via collagen provides physical resistance against these processes. Research insights continue to confirm the integral importance of vitamin C for both blocking cancer initiation and resisting advancement. Ensure daily consumption from whole food sources like camu berries and acerola cherries that provide superior absorption compared to synthetic tablets. Further boost anti-cancer effects with daily doses between 500 to 2,000 milligrams. Curcumin. The vibrant curcumin compound in turmeric root shows immense efficacy in preventing and combating cancers through diverse mechanisms. It aids cell cycle arrest. Cancer cell reproduction depends on cell cycle control, which involves several stages. Curcumin beneficially arrests cancer cells at various checkpoints within their cell cycle, effectively freezing them in place before reaching the mitosis phase, where they can split and proliferate endlessly. It causes angiogenesis inhibition. As tumors expand, they produce chemical signals to generate new blood vessels to supply their growth. Curcumin disrupts signaling by factors like VEGF, needed for this angiogenesis, helping restrict malignancy nourishment. It causes COX-2 blockage. Curcumin strongly inhibits the COX-2 enzyme, enabling chronic inflammation that encourages cancer development and related pain in those undergoing treatments. Regulation beneficially minimizes proliferative terrain. It increases apoptosis. Whereas cancer cells often shut down programmed suicide cycles, enabling their removal, curcumin both resensitizes them to pro-apoptotic signals while directly inducing apoptosis initiation, helping facilitate the destruction of abnormal cells. It helps in sensitizing chemo or radiation. Multiple studies, including a Vanderbilt University trial presented at the 2017 AACR conference, confirm that combining curcumin with conventional treatments enhances cancer cell sensitivity and the anti-tumor effects of chemo and radiation therapies without adding toxicity. It aids in DNA protection. Curcumin guards cellular DNA from damage incurred by toxins and free radicals that can spark mutations, advancing malignancy. Its antioxidant activity intercepts reactive compounds before they distort genetic material. In lung, breast, colon, pancreatic, and ovarian cancer cell research, curcumin continues to demonstrate exceptional versatility in blocking signals cancers need while urging their cell death across early and advanced stages. Therapeutic doses between 1,000 to 4,000 milligrams daily of enhanced bioavailable BCM95 extract potentiate anti-cancer effects. Pair with oil and pepperine to amplify absorption. Green tea extract. Abundant research highlights green tea extracts multi-target mechanisms for impeding cancer development and proliferation. The most acclaimed is its catechin EGCG, which demonstrates extensive efficacy. As a robust free radical scavenger, EGCG intercepts DNA damage, enabling mutation and uncontrolled replication, instigating cancer genesis. This safeguards genome integrity. EGCG blocks the COX-2 enzyme responsible for proliferative inflammatory cascades while also enhancing inherent cellular antioxidant systems to resist related oxidative damage. This compound also counteracts chemical signals cancer releases to generate internal blood supplies, feeding expansion. Restricting nutrients strangles growth. Cancer cell immortality depends on the telomerase enzyme maintaining protective caps on chromosome ends that shorten with each division. EGCG removes this advantage by directly binding and suppressing telomerase action only in malignant cells. Cancer survival partly involves circumventing normal checks and balances regulating cell cycle reproduction stages. As highlighted in OncoTarget, EGCG beneficially dysregulates phase durations and cyclin-dependent kinases that malignancies rely on. Research also demonstrates EGCG urges cancer cell apoptosis through both extrinsic immune receptor pathways and intrinsic mitochondrial signaling, as highlighted in the Nutrition and Cancer Journal. Green tea's matrix synergistically supports EGCG mechanisms via feflavins, L-theanine and polysaccharide content, which are interdependent for bioactivity and absorption. Daily consumption reduces recurrence risk and restrains proliferation by up to 50% for hormone-related cancers. Matcha varieties offer greater catechin content, approximately 100 mg per cup. Standardized green tea extract boosts this to 400 to 500 mg daily. Selenium. This essential mineral fights cancer through several key mechanisms. Selenium is pivotal for glutathione activity, and liver enzymes detoxify carcinogens before DNA damage can trigger uncontrolled replication. Deficiencies severely weaken clearance capacity. Selenium supports both the production and cytotoxicity efficacy of natural killer cells, and macrophages target cancerous cells for removal before they multiply. Supplementation strengthens surveillance. Its antioxidant capacity indirectly protects cell line DNA from mutations. Selenium also enables systems to identify damaged sites for cleanup before permanent distortion sets in, enabling malignancy. Optimal selenium levels facilitate precise redox signaling that maintains balance against unchecked proliferation related to chronic oxidative stress. Research confirms selenium supplementation restricts microvessel formation tumors required to nourish expansion. Limiting malignant sustenance helps deter proliferation. Studies confirm these collective mechanisms provide chemopreventative advantages, lowering cancer incidence and resisting advancement for varieties like prostate, colon, and bladder that are especially redox and detox dependent. However, determining need requires testing given selenium's narrow efficacy range before toxicity potential. Most adults tolerate 100 to 200 micrograms well for anti-cancer purposes. Modified citrus pectin. Modified citrus pectin, or MCP, provides a novel means of resisting metastasis, the process by which cancers migrate and then reproduce elsewhere, devastating mortality odds. MCP blocks galectin-3, an adhesion protein that malignancies bind as a sticky handhold to travel through vessels and penetrate new tissue environments. Animal and early human research shows MCP acts as a lookalike decoy, saturating available galectin-3 sites. This leaves none accessible for circulating, metastatic cancer cells attempting to latch on and spread. University hospital trials presented in alternative therapies in health medicine highlighted MCP stopping prostate cancer proliferation by inducing apoptosis after cells detached sans galectin-3 tethers. Without securing travel passage, separated cells perish. MCP also chelates heavy metals and positively charged toxins that provide nourishment and growth factors that malignant cells thrive on. Sequestering these compounds away from tumors while evacuating residual waste byproducts further weakens their acquisition of expansion elements. Ongoing research continues to confirm MCP's antimetastatic and chelating mechanisms for battling melanoma, breast, colon, lung, bladder, and additional cancer varieties. Using modified citrus pectin capsules delivering 6 to 10 grams daily supports prevention and restraining advancement by blocking galectin adhesion proteins tumors exploit. Medicinal mushrooms. Medicinal mushrooms like reishi, cordyceps, lion's mane, shiitake, and maitake demonstrate compelling anti-cancer actions through diverse bioactive compounds. Complex structural carbohydrates like beta-glucans directly stimulate immune cell production, activity, and efficacious tumor infiltration for selective malignant cell destruction. MHC complexes are also prime responses. Triterpenoids like ganideric acids modulate inflammation to optimal levels, discouraging proliferation while enabling efficient immune cancer cell targeting. Defense regulation prevents overreactions that enable disease. Compounds like coriolan restrict vascular endothelial growth factors that cancers utilize to expand internal blood supply, nourishing growth. Limiting nutrients suspends expansion. Bioactives also beneficially interact with gene expression related to tumor suppressors, proliferation factors, invasion capacities, and cell cycle checkpoint control. Genetic rebalancing impedes advancement. Unique carbon structures attract and sequester toxic heavy metals that provide raw materials and growth signaling for developing malignancies. Detoxification starves progression. Research increasingly confirms mushrooms' capacity for modulating immunity, gene expression, starving the neovasculature, and detoxifying to thwart cancer without disturbing healthy cell mechanisms. Cordyceps, turkey tail, and ricin merit particular attention for their broad efficacy. Omega-3 fatty acids. Omega-3 fatty acids, especially inflammation-resolving EPA and DHA varieties, assist gene expression while fueling responses unfavorable to malignancy. Omega-3 incorporation enhances fluidity, allowing for receptor mobility and sensitivity to signals related to apoptosis, leukocyte activity, and cell cycle regulation. Dysfunctional, rigid membranes contribute to unchecked growth. Omega-3s increase anti-inflammatory prostaglandins that counter signals enabling cancer cell survival, angiogenesis, and motility. Malignant terrain amplification is resisted. As highlighted in the Nutrition Journal, omega-3 intake favors genetic pathways suppressing proliferation, restricting adipokines, promoting obesity-associated cancers, and mobilizing natural killer cells actively targeting tumors. DHA also drives NRF2 signaling for amplifying cellular systems that neutralize and escort toxic reactive metabolites out of cells before they can damage DNA material, creating oncogenic mutations. Clinical research repeatedly finds higher omega-3 tissue concentration correlates with reduced risks across numerous cancer varieties by 35 to 50% while extending survival lengths by months for late-stage patients. Aim for 1,000 to 2,000 milligrams of EPA and DHA daily for ideal protective effects. Conclusion: integrating even a selection of these anti-cancer supplements significantly empowers prevention and combats malignancy progression through unique bioactive mechanisms. Allow their actions to synergize with abundant produce, phytonutrients, detox-supporting nourishment, movement, and community bonds for composite protection. Please like, share your experience in the comments, and subscribe to our channel for more insightful videos on health. Together, we are winning health.